Hello students, I am Dr. Ashwath. In this session, we're going to discuss about transgenic approaches in plants resistant to pest infestation. So before going to the transgenic approaches, we need to discuss about how fatal this insect infestation was or is. So we need to know that here to eradicate pest that causes loss in agriculture crops, you can see we are currently relied on the pesticides. So here, if you take the map of this, nearly 26%, we are spraying pesticides for the fruits. 6% is we are spraying for the cotton. 9% and 10% for the soya bean. And cereals and maize, we are spraying around 18 to 13 respectively. And here, India, you can observe that the crops, nearly 45% we are mainly sprayed on cotton. 22% we are spraying on rice and other type of vegetables here. If you take the consumption in India, if you consider that, so it will take around 6% globally. We can observe the consumptions here with respect to other type of countries. And if you take state-wise consumption of pesticides, here we can observe Punjab and Uttar Pradesh will consume lots of pesticides as compared to other states here. So whenever you use pesticides more, you already know the bad circumstances that happens in pesticide affected areas. Take example like in Kasargod district, uh, because of spraying of endosulfan, many people surrounding the village are suffering from mental retardedness because of the inhalation of those endosulfan pesticides. So many of them are suffering from cancer. Many of them suffering from other diseases because of the pesticide unscientifically sprayed on the agriculture field. So there is different strategies that is used for insect resistance. Currently, BT toxin is effectively used in different agriculture crops, vegetable crops, effectively eradicate other type of insects like they belongs to a lepidoptera or other type of families here. So if you take the strategies, these are the different type of strategies that is used to combat insect resistance. In that, BT toxin is very famous and currently applicable. RNA interference and other type of strategies are there. We'll go one by one. First one is called as BT type of resistant, BT toxin mediated resistant. So this the strategy is very effectively used in different agriculture and horticulture crops. First, this is identified in a free-living bacteria that is called Bacillus thuringiensis. This Bacillus thuringiensis is a free-living soil bacteria, gram positive, and that is identified by two scientists, Japanese scientists, on silkworm. Inside the gut of silkworm, they observe the infestation of Bacillus thuringiensis, and especially this thuringiensis produces a crystal-like protein. This is called as delta endotoxin or a cry protein that mainly kill the whatever the infection infected insect, lepidopteran insect, and then causes internal bleeding and then death happens to that insect. If you take the nature of the Bacillus thuringiensis, it is a gram positive rod shaped uh, bacteria. It internally it produces a bt toxin crystal protein. If you take the structure of the protein, it look like a arrow like structure, arrow that contains a sharp needle like structure. Whenever in alkaline conditions only, these toxins will be released out and then make a hole in a epithelial cells, the brush border epithelials of the Diptera, Coleoptera, and Hymenoptera, as well as Lepidopteran type of insects. If you take the structure of the protein that becomes delta endotoxin, it is of 17 to 130 kilodalton. And if you take the structure, it contains three domains, main domains: domain 1, domain 2, domain 3. And domain 1 is very important, that make a tube-like structure, and it will make a needle-like structure that mainly pierced into the brush border cells of epithelial cells in the midgut of lepidopteran insects, and helps in the internal bleeding and then death of the insects here. As I said earlier, domain 1, domain 2, domain 3 is very important, and then domain 1, which makes the cylindrical structure that helps in the pore formation in the insect midgut. And domain 2 and domain 3 mainly helps in the receptor recognition in the epithelial cell, and then domain 3 mainly helps in the protection of this degradation of this protein by different types of chemicals secreted by the midgut of the insects. How this delta endotoxin works here: whenever the insect larvae ingest bacteria or genetically modified plant that contains the delta endotoxin, whenever ingestion happens, so these protein along with the food will enters to the midgut of the insect. The midgut of the insect will have a pH of 7 to more than 7 to 7 to 12 pH will be there. Because of the alkaline pH, it will favors for the cry protein to express and produces this delta endotoxin in bacteria. But in plants, generally the cry protein is produced already and then expresses delta endotoxin. This delta endotoxin will be will produce and because of the alkaline pH, the protein alteration happens, and the helical hairpin is formed. This hairpin mainly appears the midgut of the epithelial cells of brush border cell of lepidopteran insects. Because of the holes happens in the midgut of epithelial cells, because of internal leakage, the insect died instantly because of the toxicity, or the internal leakage happened in the midgut of the insect larvae. If you take the steps of this mode of action, if the first step is ingestion, the plant, genetically modified plant that contains delta endotoxin is consumed by insects, and later on what happens, because of alkalinity, these delta endotoxin modified and then solubilized into the pH, and then because of 9.5 to 12 pH, the activation of delta endotoxin will be there, and which forms a needle-like structure. Whenever these needle-like structures bind to specific receptors, so that will damage the gut wall of these brush border cells of the midgut of lepidopteran insects. Whenever that leakage happens, the pore formation that causes cell lysis and death of the insects happens because of the cry protein. There is different types of cry protein is present, and here cry 1A protein is very essential. That is a primarily bole guard cotton guard this cry protein, and they effectively neutralizes this lepidopteran, coleopteran, dipteran type of insects here. And if you want to engineer this kind of cry protein, we need to search a suitable cry protein, and we need to select a BT strain which is suitable for a particular plant. And we need to isolate a gene that produces cry protein, and then we need to integrate it into a vector. We need to have a strong promoter for the expression. We need to go for a cauliflower mosaic virus type of promoter that is suitable for the expression in plant cells. After that, we need to integrate and transform these vectors into host plant. We'll use electroporation method or gene gun method. It's a direct method, or we can use indirect method like agrobacterial tuberculation mediated gene transfer. And after that, we need to select the hybrid plants where integration happens, and we'll go for screening and field testing. Later on, we approve the beta toxin in, producing genetically modified plant further. A successful story of beta toxin producing plant is Bolgard, that is first introduced by a company called as Monsanto. This Monsanto company introduced this delta endotoxin like the colors synthetic cry A1 A1AC protein in with the strong promoter called as cauliflower mosaic virus promoter into the boll gourd of cotton. If you observe this, you can see this is boll gourd cotton, this is normal cotton, this is genetically modified, and it produces the yield, good yield as compared to the normal cotton. So this successfully produce, this boll gourd successfully combat the infestation of the lepidopteran insects in European countries, but this boll gourd is not successful in India because the insects that is there in European countries is different. But here in India, because of climatic factors, these cry protein do not work well in temperate and tropical type of agriculture land. Later on, same Monsanto company producers introduce their cry AB genes into BT maize. They named as yield god from Novartis. You can observe that it produces successfully and then mitigate different types of diphtheral insects here. And again, one more company produces knockout and starling by agroe that also successfully produced and sold with different types of delta endotoxin cry proteins here. Cry 9C with starling and cry 1AB with respect to knockout type of BT maize. One more methodology that can be used for resisting the insect pathogenicity in plants is RNA interference. This RNA interference mechanism that is used whenever the pathogenic type of insects harbors onto the plant, definitely some proteins are produced. So we can enhance the protein, and you can also defend that to express this, the proteins that should favor for the insects here. That means the protein that is there in cell membrane should make a way for the insects that way from the cell into the pla- uh, plant bodies. Definitely, some protein should work. If you mitigate those kind of proteins that produce from the plants that favors for the infestation of the insects should be neutralized by a mechanism called as RNA silencing mechanism. Those genes, those proteins that favors for the infestation should be neutralized by RNA interference mechanism. So those genes will be targeted and then will produce as antisense mRNA, and then it forms double stranded RNA, and then risk complex is formed, and then nullify the action of those proteins that favors for the infestation of the insects here. This mechanism works effectively in neutralizing many vast varieties of different scale varieties of the insects in agriculture crops. And one more mechanism, one more type of strategy we can use is we can use a proteinase or protease inhibitors mediated resistance. What is this? That means, whatever insects that will mainly harbors onto the plant, it will eat up. A plant contains proteins, carbohydrates, and other type of necessary energy nutritional elements. If plant contains proteins, especially some insects harbors onto the seeds, agriculture crops that will reduce the yield. So in that case, if seed produces these proteinase inhibitors, especially this seed contain protein, and then along with that seed produces proteinase inhibitors, whenever insects comes and then consume that seeds, because of these proteinase inhibitors that is present in seed or other type of plant parts, definitely what happens, the digestive tract where the protein which is secreted like a cysteine, aspartic acid, or a melatoproteins or threonine protein, that the enzyme that is produces to digest these kind of proteins will be nullified because of the proteinase inhibitors. These are the list of inhibitors that can be engineered in the genetically modified plants to neutralize the protein digestion in the host insect. Sorry, the predator insect. Sorry, not host insect, predator insect. These insects, because of the proteinase inhibitors or protease inhibitors, it cannot digest the proteins inside their guts. Because of the starvation, these insects will die because of we genetically modified engineer a plant that produces proteinase inhibitors or protease inhibitors that will prevent the digestion of the proteins inside the midgut of the insect. And one more strategy is called as lectin inhibitors. Lectin inhibitors also one of the strategy where lectin also helps in the digestion of some sugar, fatty molecules, etc. These lectin inhibitors will bind to sugar molecules, fat molecules, so that insect midgut cannot channelize that into energy bodies. So because of the binding, so it cannot channelize, and then energy will not be produced because of the starvation by ingestion of these type of genetically modified plant that lecithin inhibitors, insect cannot survive and it will die because of the starvation. One more type of inhibitors that can be engineered into the plants is amylase inhibitors. What is amylase inhibitors? These enzymes produced by insects in digestion of the carbohydrates. The plant contains the reserve food material called starch and glycogen. These starch and glycogen should be degraded by amylase. Whenever the plant itself produces the amylase inhibitors, whenever the insect ingested the plant, engineered plant, what happens? These amylase inhibitors are there that will binds to the amylase enzyme and then prevent the digestion because of not digested, the insect will die due to starvation or indigestion because we engineer a chemical that produce inhibits the amylase activity in the gut of the insect. Even we can trigger out the production of more number of the secondary metabolites that mainly adversely affect on the insect infestation. So, some alkaloids, terpenoids, phenol, glycosides, glucosinolates, saponins, lignines, and then polycystinolenes, and then essential oils are there. These are the secondary metabolites that secreted from the plants to defend the insect infestation. Insects can cope up easily, but whenever you produce that in higher fold because of genetic engineering technology, so it become toxic to insects infestation, and then insect die due to the accumulation of the secondary metabolites inside the body. Even we will go with other strategies plus herbivore induced plant volatiles mediated resistance. That means you can observe that the green leaves and other type of plant parts will release some volatile compounds that damages the insect infestation, like terpenoids and polyphenols and phenylpropanoids and indole. These chemicals mainly has anti insecticidal properties are there. These insecticidal properties mainly gives out when you increase the expression of these chemicals in multiple folds in genetically modified plants so that it can easily defend the insect infestation. One more strategy means we'll combine multiple transgenic traits in a single plant. That means we discussed about BD toxin, we discussed about RNA interference, we discussed about the enzymes, alpha amylase, protease, etc. In single plant only, we'll target insert BD toxins, multiple BD toxins. We'll go with the stake resistant proteins. We'll go with enzyme inhibitors. Like that, we'll stake all the genes together in single plant so that the plant genetically engineered plant with multiple genes will have resistant in multiple ways, so it become a very good plant in defending multiple infestation. One more strategy we can use is chitinase mediated resistance. That means the chitinase enzyme which breaks down the component of chitin in insects. Whenever we modify a plant, genetically engineer modified plant in such a way that this plant produces chitinase enzyme. Whenever chitinase enzyme is consumed by the plant, consume the plant part, along with that chitinase enzyme also enters to the lepidopteran insects. What happens is chitinase enzyme degrade the chitin component of the insects. So what happens? These insects, because of internal gut leakage, it will die because of the consumption of chitinase inside the gut. One more strategy we can effectively get rid of insects is pheromones. Insect pheromones, we can use it either in agriculture crops or either in weed plants. So whenever these pheromones are secreted, insect related pheromones are secreted in weed plant or in agriculture crop, definitely distraction happens, and then there is no mating, there is no propagation, and then the plants, so insects will die due to the distraction happen. Though the energy that is utilized will be used for the reproduction mating will be distracted, and then no propagation of these insects will takes place, and we can mitigate the insect population. So there is different types of pheromones are there. Sex pheromones that attracts the opposite gender, and aggregation pheromones that mainly helps in the aggregation of insects in one place. Alarm pheromones are there that mean that will give signal for any other threat. I know trial pheromones are there that mainly helps in the interaction in guiding the insects in particular places. Territorial pheromones are there. These pheromones are secreted by individual insects to decide its territory to repel out the opposite type of insects. One more pheromone plus queen pheromone that is produced by queen bee to maintain the social order in the colony. Like that, some pheromones are there. If you target, what are the strategies we can do that in with respect to insect pheromones in genetically engineered plants to combat the insect infestation? Means you can see we can disrupt the matings here. So we'll engineer a plant in such a way that it produces a mating pheromones. So in especially in weed plants, what happens, instead of mating to female insects, they will go and binds to the weed plant, so that there is no mating, there is no propagation, and then that will disrupt the sexual cycle of these insects. Even we can go and trap the opposite gender pheromones into the same weed plants. So what happens? They will capture the insects in large percentages that will prevent the reproduction. Even some pheromones will repel back the insects, will integrate the genes into the actual agriculture crop, so that these, because of the secretion of these repellent pheromones, it cannot insect cannot binds to the agriculture crops, and then the agriculture crops will be saved because of the transgenic technology. So like the different strategies are there, we can use it. And even we can stake the cry toxins, whatever the cry different varieties of cry proteins are there, that will stake in a single gene into a single plant, host plant. So whatever, we'll stake all the cry proteins into a gene, and then with strong promoter insert in a single plant. What happens? It will have a defense with this genetically engineered plant will produce a defense mechanism against vast variety of broad range of insects like lepidopteran insects, coleopteran insects, and then other type of insects will be there. So because of that, because of taking these the cry genes entirely into single plant will leads to the broad spectrum, neutralizing the broad spectrum of insects that belongs to coleoptile, epidopteral, or even we can neutralize the nematode also. Even we can go with other strategies plus inducible defense mechanism, like will enhances the plant pheromones or hormones like jasminic acid or salicylic acid or ethylene will be enhanced more, or even will engineer the plants in such a way that they produces secondary metabolites more. Even will enhances systemic induced resistance. Even we can enhances reactive oxygen species. Even will break the physical. We can enhances the physical barrier, like the cell wall composition of the plant will be changed, so that there is no infestation of insects inside the cell or a part of the plant. Even we'll go with other strategies like we'll genetically modify a beneficial microorganisms. So not non-symbiotic beneficial microorganism that is present in the soil is genetically modified in such a way that they will also produce a toxin to mitigate the infestation of the insects or insect eggs, like in a trichoderma and pseudomonas and other type of bacteria is there, that they will be engineered in such a way that they will also produce as effective cry protein to mitigate the infestation. So one more method we can use it called as CRISPR-Cas9 technology, where in CRISPR-Cas9 technology is precisely target the insects like female related genes, male related genes, or insecticidal resistant genes, or pheromonal genes, or segmentation of genes, or metabolic genes, or phenotypic genes will be targeted, so that we can easily mitigate the infestation of the insects into the agriculture crops. So different strategies are there. We need to focus, and then we need to mitigate the infestation of insect by different transgenic strategy that is discussed in the previous slides. So here you can see, to mitigate Drosophila, we'll insert a PGSID gene, and to mitigate the beetle, potato beetle, we'll go with vestigial gene. If you want to mitigate cotton bollworm, we'll go with CYP6A gene, and even we'll go target the fruit fly, Drosophila like fruit fly. We can go with other type of receptor genes, which will mitigate by CRISPR-Cas9 technology. So like this, different strategies are there in transgenic plants to mitigate in different way, and to mitigate in different ways the insect infestation in agriculture crops.